Hi, thank you for coming to see this talk or staying where you are and seeing this talk. I'm here to provide some microbial eukaryotic perspective to this issue of biodiversity and taxonomy in an era where we're understanding biodiversity on a new scale. And I wanted to start off by talking about this paper, Eukaryotic Plankton Diversity in the Sunlit Ocean. This is a paper that looked at biodiversity from the Terra Oceans project. I was not involved with this paper, but it was sort of a landmark in terms of understanding the ocean and the smaller stuff that lives in there that is still eukaryotic. And I wanted to just show you a couple figures. So for instance, you can see here, if you look at this blue line here, this is a measure of OTUs that they were able to identify using a DNA barcode. And that's the blue line, the dark blue line is the overall. And they estimate about 110,000 microbial eukaryotic species showed up in their samples. But what was really fascinating from perspective of somebody who's worked on protists for a long period of time is in fact this blue line here, which is represented here in this OTU table. This is the sort of what they call the pico-nano size, which is between 0.8 and 5 microns. So this is a really small size for eukaryotes, but you can see that the OTU diversity is enormous in this size range. And that was something that was a bit of a shock. It's not just the number of OTUs, but in fact, if you look at, this is a breakdown of each one of these size classes and how they were classified among the eukaryotic groups. And there's a legend at the bottom that describes each what the abbreviations are, but we have a huge amount of diversity in this small size class. It's not just one group of organisms, which is really diverse. These things come from across the eukaryotic tree, which was really fascinating. Included in that are some things like this white box, Incertae sedis. These are things that we don't have a good home for in terms of their taxonomy. And then some other groups like, for instance, I'll get to this malve group in just one second. So I'm going to show you one more figure from this paper. It's a little bit overwhelming, but you can take a look at the backbone here is essentially a representation of the eukaryotic tree with all the major groups. There's a few things up top like OTU number and the number of reads that they were able to produce based on their data. But one thing that I want to point out from this tree is that there are major clades here. For instance, okay, here are these malves that I talked about earlier. Okay, there are major clades which we didn't really even know existed before these data. And it turns out that they're not only really diverse, but they're really prevalent. You can see in some of the OTU numbers here, okay? So we have this DNA data. So this is very similar, I think, to what's happening in the prokaryotic community, where we're able to identify biodiversity based on the DNA, but we don't have a good face to put to this DNA, right? We don't know what the morphology looks like. In this case, they've inferred the trophic levels. So these are in red are parasites, in green are photosynthetic organisms, and in black are heterotrophs. And so they've inferred their function, but we don't actually have any morphology for a lot of these lineages that turned up in this small size class. Now, in order to kind of understand and deal with the taxonomy of eukaryotic microbes, every 5 years or so, a large group of taxonomic experts get together and try and come up with a working classification. Now, there's problems from our perspective in trying to work together to make a coherent taxonomy across the eukaryotic tree, because, for instance, there are two codes, the botanical and zoological code, that govern how taxonomy is described. And in the cases that many of us are dealing with lineages that have some groups that are described under each of the different codes. In some cases, we have groups that are assigned taxonomy, overlapping taxonomy from different codes, which is hugely problematic. There's an example I've highlighted here, but let me just show you what that kind of looks like. So if we take this as a representation of the eukaryotic tree, and these are sort of all the major lineages, it's relatively up to date. We have the botanical code, which was designed to deal with this small group of organisms here, relative to the rest of the group, rest of the eukaryotes. But it's applied to anything that's photosynthetic, and also as sort of a bonus, it's also applied to fungi, which are down here, and that's for historical reasons. So we have one code of nomenclature, which is sort of used to assign taxonomy in a variety of different groups, because photosynthesis has moved across the eukaryotic tree in an uneven fashion. So it's applied to a broad swath of organisms. And then we have the zoological code, which again was designed for a small branch of the tree here, but is applied to non-photosynthetic organisms across the tree. Now we have instances where, of course, we have overlap. For instance, the omycetes, which are over here, this is water molds, and were responsible for the Irish potato famine. They were originally classified as fungi, so they were originally classified under the botanical code. And it wasn't until later when DNA data indicated that they're actually members of the stromatophiles. They were moved to a group that's largely classified by the zoological code. So again, we have these problems. We have similar problems to the prokaryotic community in the sense that vouchering is problematic for us. So these codes were designed for animals and plants, which are, generally speaking, large enough to be easily vouchered. When it comes to eukaryotic microbes, that's not so much the case. We don't need to actually provide cultures into multiple culture collections like the prokaryotic community has decided to do, but we still have problems with the vouchering. As the sort of broad scale taxonomy and relative relationships change because of our better understanding of the evolution of these organisms, we then get conflicts that are created between the codes. Now, in addition to all that, of course, the botanical code and the zoological code are governed by bodies that are focused on those groups, and the motivation to change the code to sort of deal with the needs of the protist community or the microbial eukaryotic community is not very high at times. So what have we gone to? So we moved to a different, bit of a different system, and this was introduced in this 2019 taxonomic framework. And essentially what we've tried to do is form a functional framework that is taxonomy for eukaryotes. And we're taking a forward-looking approach by involving DNA data and computational resources. What that looks like is essentially, so we have what's a project called the UniUke, which you can see in the middle here. And this is based on a taxonomic reference data, which is large-scale sequence data sets, phylogenomic data sets. That's housed under UkeRef. A UkeBank is a home for broad-scale metabarcode data globally. And then the taxonomic experts, which have expertise in individual groups. We combine all of that and into an output that we're calling UkeMap. So UkeMap is essentially a informed taxonomy that is a community taxonomy that's agreed on among the community. It's a centralized hub to sort of keep all, as taxonomic changes happen, they can be changed in real time by members of the community. We've got a group of taxonomic experts who sort of, I don't want to say police the taxonomy, but in essentially as changes are proposed, there are decisions made among that community. And these data will then be ported to EMBL and NCBI and DDBG and other reference databases like Silva. And so the idea here is to form a community-based taxonomy that is informed by data that are sort of ongoing all the time, and use that information so that we have an up-to-date taxonomy that can be referenced at any point. All right, so thank you. Hopefully that's helpful in thinking a little bit about the problems that the prokaryotic community has with the ongoing taxonomy, and I'm happy to answer any questions in the workshop. Thank you.